Want to first double check if you can hear me clearly and if you can see the blackboard and the PowerPoint here on your screens. All right, great. So today we are starting our discussion of the nervous system. If you remember, when we discussed the organ systems, we mentioned that the two control centers that we have in the body are the nervous system and the endocrine system. And the main difference between the two is that one is much faster, much more specific, conducting electrical signals to the target organs, and this was the nervous system. And the other one, the endocrine system, was much slower and less specific in targeting specific organs. So it's going to be sending out the message to all the organs of the body because I'm sending out chemical messages through the bloodstream. So all the organs going to be receiving this message. Only the ones that will have receptors for those hormones are the ones that will be responding to the signal. So in the nervous system, we have three main functions. The first function of the nervous system is its ability to receive sensory input. I am capable to recognize the presence of what we call a stimulus. And a stimulus is simply a change in the environment. So if you hear a sound, this is a stimulus, and this is a change in the environment. So you've got waves that are transmitted through the air, and this is considered a change of the environment that will cause the formation of a sound, and in this case, it's going to be considered a stimulus. You see something, so you have waves of photons, and those photons are going to be considered the change of the environment that allows you to perceive vision. So this is going to be considered a stimulus. So all the sensations are actually resulting from a stimulus, and this stimulus is the change of the environment, either an external environment, either something outside or something inside, the internal environment. So if, for example, I did have an increase in my blood pressure, I have the ability to detect this change in the internal environment for me to be able to respond to it. So your ability to detect the change of the environment requires you to have sensory receptors. So if I'm touching something, I need to have touch receptors. If I can't see, I have to have light receptors. If I can hear, I need to have hearing receptors, and so on. So it requires me to have receptors to detect the change in the environment, which is the stimulus. And what would I do with this signals that I've received from my receptors? I will integrate this sensation with what I already know, with other information in my central nervous system in the brain. And this is going to be the second function of the nervous system, the integration of the information that you are receiving with what you already know, with what you can perform. You can respond to the stimulus. So if you touch a hot surface, for example, you've got sensory receptors that will detect you that you've touched this hot surface. This information will be sent to your brain, will be integrated with a lot of things inside your central nervous system. And as a result of the integration inside the central nervous system, you're going to be responding. You're going to be withdrawing your hand, pulling your hand away from this hot surface. You're going to be performing a motor output. So you're sending out a signal to your effector organs, in this case is going to be the muscles, for you to respond to this stimulus. So the motor output is simply the activation of the effector organs, either muscles or glands, to produce a response. So I've seen a cup of water. This is the stimulus. I am able to detect that there is a change in the environment, that there is a cup of water present by the photoreceptors in my eye. Those are the receptors. And those receptors are going to be sending out sensory input to my brain. And inside the brain, I would have the integration between the sensation and my memories, my motor centers in the brain. And finally, I will be responding. I'm sending out a motor output to my biceps, to my arm muscles for them to contract and relax in a sequential pattern for me to grab this cup of water and drink. All right, so you've got sensory input, you've got integration, and you've got motor output. Those are the basic functions of your nervous system. We divide the nervous system into two major parts anatomically. Those are the central nervous system and the peripheral nervous system. The central nervous system consists of the brain and the spinal cord. Compared to the peripheral nervous system, it's going to be formed by the nerves going in and out of the central nervous system. Those nerves might be coming in and out from the brain, so we call them cranial nerves, or they would be going in and out of the spinal cord, so we call them spinal nerves. Those are going to be responsible here for carrying the signals, the sensory signals toward your central nervous system, and carry the motor signals from your central nervous system to your effector organs, either muscles or glands. We classify the nerves according to the type of signals they will be conducting. And accordingly, we're going to be setting up different types of nervous system. So the peripheral nervous system consisting of the different nerves. Some of those nerves are going to be conducting sensory signals. What I mean by sensory signals, I'm conducting these signals from the receptors towards my central nervous system, my brain and my spinal cord. And I have the motor nerves that will be conducting the signals from the brain and spinal cord to the effector organ. So here, I am sending from the receptor to the central nervous system. The motor nervous system or the motor nerves are going to be conducting the signals from the central nervous system to the effector organs, either muscles or glands. If I am dealing with a sensation that is detected from the internal organs, from the visceral organs, I have got a stretch in my blood vessels, I have a stretch in my stomach, I have a change in my blood sugar level. So those sensations are going to be sent through a type of nerves that we call the visceral afferent fibers. If I'm sending out the signals from the receptor to the nervous system, they are afferent fibers. So This might be coming from the visceral organs, my internal organs. So we call those fibers that will be conducting those sensations are my visceral afferent fibers. And I might be conducting those sensations from the receptors in my skin, muscles, and joints. So we call those fibers conducting the touch sensations, the pressure sensations, the vibration, the pain, temperature, all those sensations detected by the receptors located in your skin, muscles, and joints. Those are going to be conducted through the somatic sensory nervous system, or the somatic afferent fibers. For the motor or the efferent fibers, those also going to be classified according to the signal that will be sent to what target organ. So if my target organ for the motor signal, it's a skeletal muscle, so it's under voluntary control. I can control it. I can decide to contract and relax my skeletal muscle. It's under voluntary control. So we call this is the somatic motor nervous system. And if I'm sending out signals to the cardiac muscles, the heart muscles, to my smooth muscles located in, if you remember, the wall of all hollow organs, except for the heart, and to my glands. So you see, all those are not under voluntary control. They are under involuntary control. So you don't have the ability to decide, for example, to increase your gastric secretions. It's a glandular secretion. To increase your, for example, the heart rate, to increase the frequency of the contraction of your stomach. So all those are not under voluntary control. They are under involuntary control. So we consider this as the autonomic nervous system. So those fibers that would be sending out the signals to my cardiac muscles, smooth muscles, glands, those are part of the autonomic nervous system, compared to the motor nerve fibers that would be sending out the signals to your skeletal muscles. Those are considered part of your somatic nervous system. So again, how did we classify the nerves conducting the signals? According to what kind of signals am I conducting through those fibers? If I'm receiving signals, this is going to be considered a sensory nerve fiber or an afferent nerve fiber. We classify those afferent nerve fibers according to where are you getting the signals from? Where did you get the sensation from? Are you getting the sensation from your internal organs, from the visceral organs? This is going to be a visceral afferent fiber. If you're getting your sensory input from the skin, muscles, or joints, this is going to be a somatic sensory nerve fiber. The other category of the nerves is going to be the motor nerve fibers, or the efferent nerve fibers, that will be conducting the signals from the central nervous system to your effector organ. And remember, the signals that will be sent to your skeletal muscles, those are under voluntary control. So you going to be classify them as somatic nerve fibers, compared to the ones that will be sending out the signals to your heart, to your smooth muscles, to your glands. Those are under involuntary control and will be considered as part of the autonomic nervous system. We've got two divisions of the autonomic nervous system. Those are the sympathetic nervous system and the parasympathetic nervous system. Sympathetic nervous system is going to be activated in case of fight or flight, in case of stress. So what do you think is going to be the effect of the activation of the sympathetic nervous system on your heart rate? If you're fighting, what do you think is going to be the effect of the sympathetic nervous system on your heart rate? It increases my heart rate. And what do you think is going to be the effect of the sympathetic nervous system on the gastrointestinal tract, secretion, and motility? Defecation and micturition. If you are in a fight, would you feel that you want to go to the restroom? So would you tell the person you're fighting with, just give me 1 second, I will go to the restroom and then we'll come back to complete our fight? So no, sympathetic nervous system actually is going to be inhibiting the gastrointestinal tract motility and secretions. The same concept, what's going to be the effect of the sympathetic nervous system on your bronchi? Would you dilate the bronchi of your respiratory tract to get more air, or would you constrict the bronchi to reduce the amount of air? Sympathetic nervous system is going to be dilating my airways, so bronchodilation and so on. So you see on here, this is going to be the effect of the activation of the sympathetic nervous system in case of fight or flight. The exact opposite of what's going to be conducted by the sympathetic is going to be conducted by the parasympathetic. So parasympathetic is going to be activated when you are resting. So what do you think is going to be the effect on my heart rate, the effect on my blood pressure if I am resting, reading after a meal? What's going to be the effect on my heart rate? What's going to be the effect on my blood pressure? They will go down. What's going to be the effect on the gastrointestinal tract motility and secretion? If I'm resting, what do you think? Is this a good time for me to send blood out to my gastrointestinal tract to help with digestion? Is it a good time to go to the restroom if I'm resting, not fighting? Yes. So the gastrointestinal tract motility and secretions are going to be boosted by the activation of the parasympathetic nervous system. So you see on here, this subclassification of the autonomic nervous system into sympathetic and parasympathetic, it's a functional subdivision. Functional subdivision. Both are sending out signals to the same exact target organs, but they have two opposing effects. And this will allow you to regulate your heart rate according to your needs, to regulate your blood pressure, to regulate your blood sugar level. All those stuff are going to be kept balanced within their normal range by activating and deactivating the sympathetic and parasympathetic nervous system. All right, so again, a quick overview of the nervous system. We classified the nervous system anatomically into two major parts: central nervous system, which is formed by the brain and the spinal cord, and the peripheral nervous system, which is going to be formed by the nerves moving in and out of my central nervous system, carrying signals from the brain and spinal cord to the target organs, or carrying from the receptors to my central nervous system. So we classified those nerves into two major divisions. Those are the sensory nerve fibers and the motor nerve fibers. Sensory nerve fibers can be conducting signals from your internal organs. We call those are visceral sensory nerve fibers, and they can carry signals from your skin, skeletal muscles, and joints, and we call those are somatic sensory nerve fibers. Efferent division or the motor division, I will be sending out motor signals to the effector organs, and those effector organs, if they are skeletal muscles, so they are under voluntary control, we classify the nerve fibers as somatic motor nerve fibers, and we consider this as the somatic nervous system. If I'm sending out signals to smooth muscles, cardiac muscles, and glands, I don't have any voluntary control on those ones. So those nerve fibers are considered visceral motor nerve fibers, and we classify those nerve fibers as autonomic nervous system. We've got two subdivisions of the autonomic nervous system, and those are two functional subdivisions. They are targeting the same effector organs, but they have two different effects on each of them. All right, sympathetic nervous system that will be activated in case of fight or flight, and the parasympathetic nervous system, which is going to be activated in case of rest to conserve energy. So our first in-class activity question for today: while you're studying for an exam, you reach for a beverage, you extend your arm, your blank nervous system is activated. So which nervous system would you activate to extend your arm? Is it sensory or motor first? Is this a sensation to extend your arm? Is it a sensation or is it a motor action? Motor, right? Is this voluntary or involuntary? Did you decide to do it or you find yourself doing it? It's voluntary, it's under voluntary control. So you decided to do it. So what do we call the nervous system that will be sending out motor signals to the skeletal muscles? What do we call this one? It's remember, this is my somatic nervous system. Somatic nervous system. So the good answer on here is going to be the somatic nervous system. All right, our second question. So the door slams shut loudly and you flinch. After a few seconds, you realize that your heart is beating very rapidly and forcibly. This response is a result of your blank nervous system. So what happened here? What happens in here? The heart is beating fast. So heart is beating. Is this a sensation or a motor signal? Here I'm asking, what is responsible for your heart to contract, not for you to feel that your heart is contracting? All right, so here, this response, what is the response? Is that my heart is beating. So my heart is beating. Is it motor or sensory? For the heart to contract, is this motor or sensory? Contract, muscle contract. Will it be motor or sensory? What do you think? Muscle contract. muscle contract. Remember, if I am enhancing the cardiac muscles to contract, smooth muscles to contract, or glands to secrete, all this is part of my motor, remember? So my heart is beating fast. So is this motor or sensory again? One more time. It's motor. So who's going to be responsible for the heart to contract? Is it somatic or autonomic? So for the skeletal muscles to contract, it's somatic, it's under voluntary control. Did you decide to increase the heart rate? No, you did not. It's under involuntary control. So what part of the nervous system is going to be responsible for this? It's going to be the autonomic nervous system. And something that we just mentioned, which part of the autonomic nervous system is the one that would increase my heart rate? Is it sympathetic or parasympathetic? Which one increases my heart rate? Sympathetic. So you see on here, what's the good answer? You've got afferent. Afferent, it's not specific. It's motor. Yes, it's motor. But motor afferent might be somatic or autonomic. Central, somatic. I'm not sending out signals to my skeletal muscle for it to be somatic. I'm sending signals out. It's afferent, yes, but I'm sending those signals to my heart. So it's motor and it's under involuntary control. It's autonomic. So this response, what we mean by this response is that the heart is beating very rapidly and forcibly. All right. Any questions? Any questions? All right, so what kind of cells are forming my nervous system? I have got a major type of nervous system cells. Those are the most important cells responsible for the conduction of the electrical impulses. They are long-lived cells. They can live for 100 years. Those are the neurons. They are highly specialized cells that will be responsible for the conduction of the signals. And those neurons are amitotic. What do we mean by amitotic? They cannot divide. They are permanent cells. So you cannot regenerate. If a person lost a brain cell, it cannot be replaced. And we've got supporting cells, or what we call the glial cells or neuroglia. Supporting cells, if you remember, when we studied the tissues, we've seen the neurons. Those are the cells with many processes coming out. And we've seen in the background of those nerve cells, many nuclei. And we mentioned at that time that we're going to be discussing them in details when we get to the nervous system. And here we are. Those are the neuroglial cells. So first neuroglial cells that we're looking at today, it's the astrocyte. From its name, what do you understand just from the name? Astro means star. Cyte is cell. So this is the cell that looks like a star, as you see here on the diagram. It serves mainly to connect the blood vessels to the nerve cells. Those astrocytes are going to be responsible to regulate the energy metabolism and to transport the nutrients from your blood to the nerve cells. And because those neurons are highly precious, I cannot regenerate them. So I need some kind of barrier to be located between the blood and those nerve cells. So what acts as a barrier here are also the astrocytes. They act as part of my blood-brain barrier to prevent any toxins from reaching easily the nerve cells from the blood. So if a person got a toxin in his blood, the first cells that will get affected are the astrocytes and not the neurons or the nerve cells. Think of it this way. So neurons are highly specialized cells. They cannot regenerate. They are very hard. They work very hard to allow the communication to take place between your internal organs. So you need some kind of protection. Try to imagine the nerve cells are like a CEO of a very large company. So they don't have time to cook for themselves. So they will hire somebody to cook for them. Those are the astrocytes. And they will hire a bodyguard because they don't want to have a direct exposure to the public. So again, the astrocytes here will be cooking for them, will be preventing any blood-borne substance from reaching those nerve cells directly. Another cells that we're looking at in here, it's the microglial cell, and from the name microglial cells, those are tiny little cells. And those are defensive cells. They are the macrophages living inside the brain. They are ready to fight any bacteria that or viral infection that gets into the nervous system. So the microglial cells are the defensive cells of the central nervous system. Another type of neuroglial cells that we're looking at are the ependymal cells. And the ependymal cells, as you see on here, those are cells with cilia and they would be responsible to allow the propulsion of the fluid inside your brain cavity. So inside your brain, you've got cavities, and we call those cavities are my brain ventricles. We've got 4 ventricles. This is how they look like. And as you see on here, this cavity is also going to be connected to the space around the brain. So inside those ventricles, and a canal that will travel inside your spinal cord and around your brain and spinal cord, you keep a fluid, and we call this fluid is my cerebral spinal fluid. or CSF. In order for the CSF to circulate, we don't have a pump that would actually be pushing the CSF for it to circulate inside those brain ventricles and around the brain and spinal cord. But inside those spaces, we've got the ependymal cells with their long cilia, and those cilia will allow you to pump and push the CSF to circulate within the ventricles and around the brain. So the ependymal cells on here would be lining the cerebral spinal fluid filled cavities to allow its circulation, as we don't have something to pump this fluid for it to keep moving. Another type of neuroglial cells that we're looking at today, those are the oligodendrocytes. Oligo means few. So if I am looking at a sugar that is formed of a single unit, we call this is a monosaccharide. If I'm looking at a sugar with 2 units, it's a disaccharide. If I'm looking at a sugar with many sugar units, it's a polysaccharide. And if I'm looking at a sugar with few sugar units, it's called an oligosaccharide. Oligo means few. And if you see on here, those cells will have few processes coming out. That's why we call them oligodendrocytes. Oligo means few, and dendro, it's a process, and cyte is cell. So those are the cells with few processes coming out. Oligodendrocytes. Those cells are going to be located in the CNS, in the central nervous system, and they are going to be responsible to form the myelin sheath around the nerve fibers. So those yellow parts on here, those are the nerve fibers that will be conducting the signals. You are wrapping those nerve fibers by myelin sheath, and the cells responsible for the formation of the myelin sheath inside your CNS are the oligodendrocytes. The cells responsible for the formation of the myelin sheath around the nerve fibers, but this time in the peripheral nervous system, outside of the brain and spinal cord, those are called my Schwann cells. So a quick recap on here. What are the different neuroglial cells? We've seen the astrocyte that will be responsible for the energy metabolism and to act as a blood-brain barrier, to act as a barrier between the blood vessels and the nerve cells. The other neuroglial cells that we've seen was the microglial cell, and this is a defensive cell located inside the CNS. Seeing the ependymal cells, and those are the cells with cilia lining the cerebrospinal fluid-filled cavities inside your brain, which will allow the cerebrospinal fluid to circulate within those cavities. Oligodendrocytes, those are the cells responsible for the formation of the myelin sheath that will be wrapping the nerve fibers inside the CNS. Schwann cells, on the other hand, they are forming the myelin sheath, but remember, those are forming the myelin sheath around the nerve fibers outside of the CNS in the peripheral nervous system. Looking here at the structure and the characteristics of the nerve cells, the neurons. Neurons are, again, long-lived cells. They live for 100 years or more. They are amitotic, again, means they can't divide without mitosis. They have a very high metabolic rate, and that's why your brain requires continuous supply of oxygen and glucose. So if a person has a drop in his blood sugar level, he will lose consciousness. Why? Because simply your brain can't function if he doesn't get continuous supply of glucose. If I'm not able to breathe for whatever reason, what what would I have? I will lose consciousness. If I am in an environment where I have low oxygen pressure, I'm not able to deliver enough oxygen to my brain, and this interruption in the oxygen supply gonna cause my brain to stop working and resulting in me losing consciousness. The neuroglial cells are not amitotic. Those cells can divide. The neuroglial cells are mitotic cells, they can divide. The ones that are amitotic, those are my neurons, my nerve cells. All right. So how a nerve cell looks like, a neuron usually have a cell body, and this cell body is going to be connected to short tapering processes. We call those short tapering processes are my dendrites. Also attached to the cell body of the nerve cell, we've got a long process. We call this process is the axon. And this bulging part that serves as the attachment between the cell body and the axon, we call this as the axon hillock. And why is this specific part is highly important for us? Because this is where we've got the summation of the electrical impulse. So I've got electrical impulses on here traveling through the cell body. They will be added together in the axon hillock for you to form an effective electrical signal that can travel through the axon. The axon has terminal ends like this that looks like a tail. We call those terminal ends are my telodendria. Those are the terminal branches of my axon, because these are my telodendria. Also, at the end of each of those terminal ends, we've got a bulging part. This is the secretory part that will be releasing the neurotransmitters that will allow you to transmit the signal to the effector organ or to another nerve cell. We call those are my synaptic knobs or boutons. Also, something that we need to notice on here is that those axons are going to be wrapped with myelin sheath. two main functions here of the myelin sheath. Mention one later. The first one that we need to know, why do you think if I'm holding a wire like this, is there any electricity transmitted through this wire I'm holding? So I'm connecting here my headphones to the wire. Is there any electrical signal that is transmitted, any electricity that is transmitted from the plug, it's plugged, to my headphones on here for it to charge? You see, it's red on here, so it's charging. So is there any electricity traveling through this wire? Yes. Why this electricity is not transmitted to my hand? Because it's insulated, as you mentioned. Insulation. So the most important function here, one of the most important functions here of the presence of those myelin sheath wrappings of the nerve fibers. And when we say nerve fibers, we mean the axon. So whenever we say nerve fiber, those are axons. Why do we have those wrappings? The first reason is to allow the insulation. Again, how insulation is going to be taken place on here of the nerve fibers is by the presence of the myelin sheath. And if you remember, what cells are responsible for the formation of the myelin sheath that you have in here? What cells are responsible for the formation of the myelin sheath? In the CNS, those are my oligodendrocytes. And in the peripheral nervous system, those are my Schwann cells. So if we're looking on here at the axon of this nerve cell, we see the Schwann cells forming the myelin sheath. And something that I want you to notice on here is that the myelin sheath, it's not continuous, it's interrupted. There is an interruption in the myelin sheath. We call those interruptions of the myelin sheath wrappings. Those are the nodes of Ranvier. Nodes of Ranvier. And we're going to learn more about the function of the nodes of Ranvier after break today. All right, so again, what is the structure of a nerve cell? Nerve cell has a cell body. This is where you have the nucleus of the nerve cell. I have short tapering processes. Those are called my dendrites. And I have a long process that I'm connected to. This is called the axon. The axon is attached to the cell body by the axon hillock. Remember, the axon has terminal branches. The axon terminals, or we call them altogether the telodendria. Those nerve endings have a bulging end. We call those are my synaptic knobs or synaptic boutons. And those will serve as a secretory part that will be releasing the neurotransmitter to affect the effector organ or to transmit the signal to another nerve cell. Notice on here, again, we've got the axon wrapped with myelin sheath. And the cells responsible for the formation of the myelin sheath in the peripheral nervous system, this is what we're going to be mostly concerned with in this chapter is the Schwann cells. And you will see that the myelin sheath, it's not continuous, it's interrupted. And those interruptions in the myelin sheath is what we call the nodes of Ranvier. The axons here are acting as the conducting region of the neuron. Those will be responsible for the generation and the transmission of the electrical impulses. We call those electrical impulses that have the ability to be transported for long distances are myaction potentials. So they carry the signals away from the cell body. Molecules can move along the axon into two directions. If I am moving towards the axon terminal, this means I'm moving from the cell body towards the terminal ends of the axon, we call this as an anterograde movement. If I'm moving backwards towards the cell body of the nerve cell, this is going to be called a retrograde movement. So again, molecules can move in two directions inside the axon, either an anterograde conduction or a retrograde conduction. Myelin sheath, remember, it's formed by the Schwann cells, and what other cell is responsible for the formation of the myelin sheath, but inside your CNS? Can you remind me of the name here? Anybody? Oligodendrocytes, exactly. Oligodendrocytes. So what is the function here of the myelin sheath is going to be to protect and electrically insulate the axon. Electrical insulation of the axon for the electrical signals to not be transmitted to nearby structures and to be kept moving along the axon. Also is going to be responsible to speed up the electrical conduction. So it increases the speed of the electrical conduction, as we're going to learn after break how we do this, how it plays a role in the increase of the speed of the electrical transmission along those axons. So you see on here, the Schwann cell will be wrapping the axon and will be wrapping its plasma membrane loosely first around the axon, forming multiple layers, and then will be forcing out the cytoplasm between the membrane. So the cytoplasm is going to be moving, and the membranes here will become tighter wrappings around the axon. This forced cytoplasm is what we call the Neurons. So you see on here, I will be squeezing out the cytoplasm to the outside. So this part is filled with cytoplasm. But in direct contact with the axon, you've got multiple layers of plasma membrane around the axon. So we call the outer part that contains the cytoplasm on here, it's the Neurons, and those tight wrappings of plasma membranes in direct contact with the axon is what we call the myelin sheath. So again, what is a neurolemma? Neurolemma is a peripheral bulge of the Schwann cell cytoplasm. Nodes of Ranvier, those are the gaps between the in the myelin sheath between the adjacent Schwann cells. Some axons will be unmyelinated, so they would be thin nerve fibers. They have only one Schwann cell for each 15 or more nerve fibers, so they don't have a lot of wrapping to them, so they don't have any significant myelin sheath around them, and this will make those axons or those nerve fibers have a much slower speed of electrical conduction. We classify the neurons into three main types. We have multipolar. As you see on here, the cell body has multiple processes. Many of them would be the dendrites, and one long process, it's the axon. So the cell here has more than one pole. We call those are multipolar neurons. Other neurons might have only two processes coming out from the cell body. One will act as a dendrite, and the other one will act as an axon. We call those are bipolar neurons. So they have two poles, only two processes coming out from the cell body. Another type of neurons are the ones with just a single process coming out from the cell body. Part of it will act as a receptive region, and part of it will act as a secretory and conductive region of the cell. So we call those nerve cells are unipolar cells or pseudo-unipolar, because actually it's not unipolar, because this single process coming out from the nerve cell, it's splitting into two processes, one acting as a dendrite and one acting as an axon. Examples here of multipolar cells, those are like the Purkinje cells of the cerebellum. Cerebellum, as we're going to be studying in the next chapter, is going to be responsible for the coordination of the skeletal muscle movement, and it allows you to maintain your body balance. So in order for me to maintain the body balance, I need to receive a lot of information concerning the state of my muscles, the state of my joints, whether or not I'm touching the ground, the position of my head in the space. All this information needs to be integrated and collected in order for me to send out few signals to the skeletal muscles to determine the force of their contraction for me to be able to maintain my body balance and my muscular contraction coordination. Example here of bipolar cells, bipolar nerve cells, those are the cells of the retina. As we're going to learn in chapter 15, we got bipolar cells in the retina. They would be receiving signals from the photoreceptors, these receptors responsible to detect light, and they would be sending out those signals to other nerve cells. We call them the ganglion cells that will later on form the nerve conducting those visual impulses from the eye to the brain. Example of unipolar cells are the dorsal root ganglion cells, and those are very important in terms of facilitating the electrical conduction and the reflexes. So I don't want to have any relay of the signal inside the nerve cell. I want to have faster response. Functional classification of the neurons, those are going to be to satisfy the three functions of the nervous system that we've mentioned earlier in this chapter. So we've got some neurons that are conducting sensory impulses, so we consider them the sensory neurons. So they will be conducting the signals from the receptor to the central nervous system. We've got motor neurons. Those are the ones that will be sending out the signals from the central nervous system to the effector organ. And we've got interneurons, which are responsible to integrate the different impulses within the central nervous system. And those are responsible for the integration. They are going to be the association neurons. All right, so now the question, how do we create an electrical signal, and what is the structure of this electrical signal, and how do we conduct it through the length of the axons?